All right, everybody, we're going to get started on our lightning talks. These are talks that are 10-minute talks from our vendors and our sponsors. We're going to do their 10-minute talk, and then there's 5 of them. And so we have time between each speaker. If someone wants to ask a question, like maybe 1 or 2 questions, and me and Thane and I are going to be walking around with a microphone. So raise your hand and let us try to run to you as fast as we can. To make sure that the speaker can hear your question. Okay, with that, we're going to start with Aaron Tyznik, and he is from now what we call Waters Biosciences, formerly BD. All right, it's all yours.
All right, well, I better start the timer because I have 10 minutes and probably an hour worth of content. So I'll try to go rather quickly and let people basically know what's changed and what we're doing at BD, what our R&D teams are doing. And one thing that has changed is we are now Waters Biosciences. But besides that, not much else has changed. We still focus on our customers, flow cytometry, single cell analysis, and I'll be talking about some of the aspects of Spectrum to Insight, some of the innovations that are driving higher dimensional single cell analysis. So when you think about BD Biosciences, because we still refer to that in that context at this time, is we create instruments, we create reagents, we have informatics, and we have a ton of R&D scientists, immunologists like myself, protein chemists, engineers, trying to create pretty cool technology that's trying to answer complex questions and removing all of the challenges that we can have with our technology to basically make cleaner decisions or observations about our biological differences. And so I'll start out with reagents, right? Like we think about reagents and, about 2017 or 18, I was working with Florian Mayer, and we were doing high parameter flow cytometry, the first 28 color panel. All the way back then, we started talking about we don't need more fluorochromes, we need better fluorochromes. And we set out on an activity to say, what is it that defines better fluorochromes? Let's kind of wipe the slate clean and think about it from where the future is going, spectral cytometry, also the current install base of instruments that are available, and then what makes a better fluorochrome. And so with that, we really started obviously going to the most problematic area. You heard earlier talking about replacement of brilliant blue dyes, for example, like that was just to get us by. And like now we need better dyes. We came out with real blue dyes and real yellow dyes, addressing kind of that PE cross laser challenges that we've had. But it wasn't always about just sticking more peaks in there, right? It's like, what is going to really optimize the spectral space that's available and using smarter tools other than just me and others going, well, here's a spot where we can stick a dye in. Let's actually be intelligent about this. And this was driven by Peter Mage and other great people within our R&D teams to algorithmically try to figure this process out. And that led to a whole new family of dyes called real blues and real yellows, which are also meant to be compatible on all the instruments that are out there, right? Like we know that it's not just Waters Biosciences instruments that are going to be sitting everywhere. And so we wanted to make sure that we have that compatibility. But when we think about better, obviously we started in real blue and real yellow. And for someone who knows of, I saw Julie Hill here, the Valley of Death, as Mario used to call it. That's an area that needs a lot of improvement, right? And that's across all laser lines. And so we're taking a look at it from the perspective of, we just don't want to create a whole bunch of new dyes and then you have to buy new stuff. Like there has to be reasons why you want to use these new dyes. And so we're looking across all the laser lines, looking out in the edges, the book edges, as I used to describe it or call it, and others call it that as well, in the far reds. Is there space where we can actually get new dyes with the new detector systems in there? Where are the challenges in the 600 space and so forth? And being strategic on how we think about dyes. And a better dye is more precise excitation, less cross laser excitation, tandems that aren't getting excited by a dye, the acceptor getting excited by a laser that you're not even thinking about it, and then lot-to-lot consistency, right? If we're going to have a robust biological analysis, we need to make sure we're consistent. Limited background, we don't want cross-reactivity, binding monocytes and other things like that, working intracellularly, and not having to use all sorts of special buffers to make it work, right? You just want to stain with your reagent and then have clean data. And that's what we're focused on. And so that's where we are with our reagent perspective, and you'll continue to see new dyes coming out under the real whatever laser line name. And they're meant to also pair well with our spectral instrumentation. This is our FACSDiscover S8 and A8, our analyzer built from the ground up, a new loader. Yes, a new loader. It's great. Love it. And then, of course, the sorter as well. And when you bring these into tandem with the advanced QC approaches that we have on these instruments, like we want to be able to run a panel from one instrument to the next, across sorters, across everything, and not have to change a single thing and get essentially the same data. Well, that comes into what we've done with a lot of our internal QC, and then, of course, optimizing it with our reagents that we're developing to improve spreading, real-time QC of the instrument as it's acquiring sample, accounting for variations while you're running your samples. And that allows us to drive into the place where this was the OMIP 102 with Andrew Konechny, who many of you know here, recently with our 50 parameter panel. But the thing is, this is before we had a lot of the new real dyes as well. Like, this was possible with the technology of the instrument and the reagents, and you can go beyond it. Not saying that anyone should be doing routinely 55 color panels, 60 color panels, unless you have a very serious justification for it. But it means that you're in a space where you can do 20, 30 color panels, and they're going to be easier. And with that, with all of these new technologies that we've been building, the algorithms for this development, we put it into other places as well. For example, the BD Research Cloud has a panel designer. It takes all the information, the learnings that we have, hotspot matrices, all of these new tools that we can use and have an algorithm essentially create panels that should work for you. Now, when I look at it and it says I should be using V450, I scratch my head, but then I scratch my head again as an old school panel designer. It's like, I wouldn't choose that, but actually that's probably going to work. It's probably a smarter decision than my bias that I would put into it. And so I encourage you to give it a try. Try it out. You don't have much to lose, and you just gives you back panels, and then you're like, really sit and think about it. It's like, yeah, that's probably going to work. And so it uses all the things that we've tried to take out of all of our panel designer minds and then put it into algorithm form. But besides that, running out of time, is these instruments now, the FACSDiscover family has imaging associated with it. So it's a standard flow cytometer, and we're creating an image, but we're not taking a picture. And we aren't analyzing a picture. This is embedded data and information that comes with your flow cytometer FCS file. And every single dot, those are just parameters, additional parameters associated with your data file. And if you mouse over them, you see a cool little picture. But that's all the analysis already happened, and it's embedded in there. You get information about radial moment and center of mass or nuclear translocation and things like that. And that allows us to sort on it in real time. You can do things even like looking at your nuclei when you're doing your ATAC-seq. I showed this because single-cell multiomics, you think you put good stuff in and you get bad data out. And there's now new ways to really evaluate and look at your data, know what you're running through for your samples and what you might potentially get out. And what does it mean for your data? This is just a simple 18 fluorescent parameter panel without any of the imaging parameters associated with it. You run a Tyznik, you get these clusters. We can go in, well, is this the right way to do this or not? That's side conversation. But if you throw then all of your imaging parameters in there and then you run your Tyznik analysis, what do you get? You get a lot more clusters because you have another 12 or 20 image-derived parameters associated with every single cell that has gone through there. So next I'll go on how can you use this. So CAR T cells, you want to generate a CAR T cell. We actually have a new single stain CAR T detection reagent, one-step BCMA CAR. You can develop your CAR T cells, you can stain your cells. We have one for CD19. We look at traditional flow cytometry, and if we look at our CAR positive cells on the right, okay, yeah, we have 22% CAR positive. But if we look at those and then look at our imaging parameters over here, exact same thing, we have different profiles of staining of those CAR reagents. And that's probably a continuum most likely. Maybe they got activated more and so forth, but, we can actually say, well, is there differences in these cells, right? You could not see these. We've gone upside down and sideways to see if we could identify these with traditional cytometry, and you can't. You need this imaging to be able to do that, or the image-derived parameters to be able to do it, and you can sort them. So, of course, we have single-cell analysis technology as well, like to do whole transcriptome analysis and so forth. So, like an immunologist, I'm too curious. I have to sort these things and do something with them. And so we sorted them and used our microwell technology. And then we did our clustering analysis and just looked at differential gene expression. I'm not trying to make claims about CAR T cell generation, but there's probably something here if someone wants to look at this. And so it turns out that we have differential gene expressions. There's data about polarization or recycling of the receptor and so forth may mean something for the quality of the CAR. And this is a new space that we all get to go out and play in. And with that, I'd like to thank the amazing team that I work with. There's countless people, and I cannot get them all on here, but I'll end with that and take any questions. And my team is here as well at the booth.
Okay. Yeah. Okay, so my name is Laura Johnston. I'm a remote application specialist with Cytek, and I'm going to talk about how you can get the most out of Cytek Cloud and our SpectroPanel algorithm. And do have a little disclaimer, but I'm going to skip through that. Okay, so I'm going to talk about our digital ecosystem, Cytek Cloud. It currently houses 4 integrated online tools. So that's the full spectrum viewer, the panel builder, which has our SpectroPanel algorithm, our experiment builder, and our newest one is the quote builder. Each of these are tailored for our full spectrum profiling instrument configuration. So whether you have a 1 laser instrument or a 5 laser or anything between, you can adjust that in your count, or you can change it in any of the tools, and everything will be automatically adjusted to that configuration. You can sign up for an account at Cytek, or not Cytek, at cloud.cytekbio.com. I'm going to talk about or give you a little quick overview of each of these tools. For those of you who are familiar with it, I'm also going to point out maybe some of the newer features that maybe you missed. So starting out with our full spectrum viewer, this is where you can explore new fluorochromes. So starting out, we have the spectrum viewer that shows our normalized signatures here. We have quite a few shown. We have the similarity matrix and complexity index, so you can evaluate how 2 fluorochromes interact with each other, or how they're similar to each other, or how a whole panel is together. You can evaluate spread using either the stain index reduction, the SIR, or we also have the SSM as well. And you can also look at spectral signatures as a general reference point. So all of these tools are really helpful when you're designing a panel. And additionally, if you're also just trying to evaluate your unmixing, these are also some really useful tools for you. Moving on to the panel builder, this combines all of our full spectrum design tools in one place. So all of those tools that I just said are also located in the panel builder. There's lots of options here, so you can build a custom panel manually. You also have the option to use our algorithm, SpectroPanel. You can modify some pre-designed panels like the OMIPs. You can enter a previously used panel if you want. If you're just curious how the metrics look for something you've been using on another cytometer, that's available to you. It is a guided workflow. So if you have used our software SpectroFlow on any of our instruments, our spectral instruments, that sort of tabbed interface, wizard interface is very common throughout all of our software. So it's not only in SpectroFlow, it's also in Cytek Cloud. So starting out, the first tab is select markers. So that's going to be where you're going to enter the marker names. It's also going to auto-populate your antigen density and antigen classification. But if you want to make some adjustments to that, you can always change it. The second tab, you'll assign marker co-expression or antigen co-expression. That's super important to understand in panel design. In the select fluorescent tags tab, you're going to pair the markers to the fluorochromes, or you can also use SpectroPanel in that tab. I do want to mention something that sometimes people miss is that when you are selecting the fluorochrome, there is in that drop-down menu of fluorochromes, there are some little icons that can give you some helpful information. So if you see the little swirly Cytek logo, that means that you can purchase that antibody from Cytek. It's either part of our Cytek reagent catalog or it's part of our partnership catalog, so Thermo Fisher and BioLegend. If you see this little gray icon, that means that Cytek is aware of a vendor that manufactures this. So you should always verify that, but we think that you can buy it from somebody out there. And if you don't see an icon at all, that means that Cytek is not aware of any vendor that makes that. So either you're going to need to go find that or make a custom one. And then the last page is review and order. And so you can review all the reagents that you selected, and you can also even order from that page. I mentioned SpectroPanel. That is our algorithm that automatically pairs the markers to the fluorochromes based on the input that you've provided. So you still have to enter in those two tabs with the markers and antigen density and marker co-expression. There is also a separate place, a library, where you can enter your reagents that you have in your lab. And if you want the algorithm to incorporate that information into your panel design, that's totally possible to do. And then it will very quickly give you an output of a optimized panel. And you might be curious what that looks like. So I'm quickly going to show you a little bit of data. We took an OMIP. It's a recent 28-color OMIP publication that was analyzing gamma delta T cells. And using all of that information in that publication, we entered it into SpectroPanel and had it design a panel. And did it really quickly. I don't know the exact time, but it was most likely like 2 minutes or less to design that panel. We tested it with the original panel versus the SpectroPanel designed panel, all at the same time with fully titrated and optimized reagents. So same donor, same instrument. And this is what the data looks like. So here's the original panel. We can see all the lineage populations are really nicely defined here in all these plots. And then here's the fluorochromes at the bottom here. And then this is the SpectroPanel designed panel. You can see the data is very similar. We can resolve all of those populations really nicely. So there are lots of markers where comparing the two panels, they were either equivalent resolution or even slightly improved discrimination of the populations. There were also some markers where we found that we had improved resolution. So again, on the top here is the original, and on the bottom is the SpectroPanel algorithm data. So in the first plot, you can see that SpectroPanel assigned a slightly brighter fluorochrome, and that led to decreased spread in the negative, which can help with resolution. In the middle one, you can see originally the resolution wasn't that great, but we saw some improvements in the SpectroPanel designed panel. And then finally, this last one, we see much better definition of this population. You can see clear negative and positive, whereas in the original panel, I would say it's a little bit unclear as to whether or not the positive and negative population are overlapping. Okay, so once you have your panel, the next tool is the experiment builder. So you can set up your experiment to run it on the instrument. And so great thing about Cytek Cloud is it's used on any web browser. So if you are someone who likes to sit on your couch in your pajamas and do work, then this will be really helpful. or if you are someone that works in a core or works with a core cytometer and you're charged for the time on the instrument, you really want to make efficient use of what you're paying for. You can set this up at home so that when you show up at the instrument, you're just ready to run all your samples. So you have options here. You can either start from a totally blank template, or if you've used the panel builder, you can immediately transfer all of that panel information into the experiment builder so you don't have to retype all of your markers and floors. You also have the option to configure your acquisition settings and keywords ahead of time. Or if you don't want to do that ahead of time, if you just want to set up like the tube names and plate layouts and then show up at the instrument and make more adjustments, all of that flexibility is there. So the experiment builder layout in Cytek Cloud looks very, very close to what's in SpectroFlow on the instrument. I just want to point out one thing that I don't know how well you can see this, but here there is an import button. We had a lot of customers requesting that when they run plates, they want to be able to use an Excel file to lay out all their plates and then just import it in. So we listened to you guys and we have included that in Cytek Cloud, and it is also in the newer version of SpectroFlow. So 4.0 and above, that feature is there for you. And then the last tool is ordering your reagents. There are two places you can do this. So everything is contained in the panel builder tool. So at the end, the final tab of that, you do have a button here to be able to just quickly order all of those reagents. Or we do have a dedicated tool. So we have something called Quote Builder, and you can use that specifically to select all of your reagents and build a quote from there. So you don't have to wait on, emailing your sales rep necessarily. You can just do it yourself. And then I just do want to let you know what's coming next. This is not in Cytek Cloud yet. I don't know when it will be in Cytek Cloud, but the next thing coming is that there will be an antigen explorer that will be added to the panel builder. So if you've looked at that antigen density and classification, and you weren't sure if you liked the default, what was entered, and maybe you just went to Google and tried to figure out from some publications, or maybe you looked at PubMed and tried to figure out on your own what is the antigen density and classification for your specific tissue and cell type of interest. And you're not sure, how did they run that? What were the voltages? What was the fluorochrome? There's now going to be one centralized database that you can look at this information and you can compare things to each other. And so if maybe you're running a marker that's normally studied in T cells, and so we kind of make some assumptions in the default values, right? So we'll put what most people run, but maybe your experiment, you're looking at that marker on monocytes, and that's not very common. So you would be able to use the antigen explorer to determine that, oh, maybe there's a slightly different antigen density, and then you can make some adjustments and have that before you build your panel. So if you have any questions, I guess, I don't know if we have time to take one, or you can come see us at our table. Right.
Thank you, Michelle. Hey, my name is Tim Crawford, Senior Manager at the Informatics Group of BD Biosciences, which is now Waters Biosciences. My disclaimer is a little bit lower on the slide than Aaron's. But yeah, for all intents and purposes, we're still BD and working under that name for the near future. Today, I'm going to talk to you about FlowJo version 11, the latest version that we have come out with for analysis tools for your FCS files. And I am focusing on 11 because that's what was requested. But do know that we do have a product similar to the Cytek Cloud called BD Research Cloud with a panel builder algorithm that's just been released, and a whole bunch of tools to help you build and design those panels and get the reagents you need to get high resolution, high parameter biology insights out of your flow cytometry experiments. So the first thing to mention is that V11 is a single window. We're calling it the workbench. It's an immersive application that gives you a whole bunch of access to the different things that you would find in multiple windows within version 9 and 10 of FlowJo. Over on the left-hand side here, we've got this navigation bar that has icons. As you click on these different icons, can you see my thing? No, you do not. How do I do that? Let's see. Boom. Pen, laser pointer. Don't waste my time here. Can you see my pointer? Yes, okay. So here is a navigation bar. And as you click on these different icons, you change what's in the center discovery panel. In this case, I'm showing, a graph window-like plot. And then down below that plot, there is a graph gallery where you can change what you're viewing, whether it's by sample, by parameter, looking at ancestry or backgating there. Over on the left top corner, you've got your groups pane. That is a acquisition data group with all your files in it. Then there's a experimental data group, which is where you can subgroup all of your samples. There's a compensation group that you would focus on in order to initiate a compensation matrix calculation. And everything is contextual, right? If I click on a group here, and then a population, and then a sample, that's what I'm going to be seeing in the primary plot here. On the right-hand side, there's also a navigation bar with different icons depending on the context. This will change, but this slides out a drawer that gives you all of the properties for the things you're interacting with in the center. This works really nice on a large monitor. If you have a double wide, you can spread it out. You can actually collapse both of these sidebars and just focus on the center. Move the divider up so you can see just an entire view of paneled plots. For your discovery and phenotypic visualization purposes, it's really powerful. There are themes. If you don't like that big white view, you can go with a dark mode. I'm going to be using what's called Madison theme. They're all standardized options in there. If you focus in on the hierarchy, as I mentioned, we have groups at the top that organize you into compensation data, into experimental data, subgroups. All of the gates you create are colored based on the group that you're on when you initiate the creation of that gate. So the white indicator says that I have made all these gates at the experiment data level, and then I can sync those to that level whenever I need to. Down below, we have the samples list, similar to the workspace in version 10 and 9. You can add statistics or keywords to this list to make it a little mini table that's always present. And this is all how you navigate, right? Clicking on the group and the sample and the population you want to focus on. There is something called the metadata manager section, which replaces the inspector in version 10 and has more functionality. Here there are 3 tabs. There's a keywords tab, which just shows you all the keywords that are in the files. You can pivot this keywords table. You can search within it. You can add your own unique keywords and then label them to parse out your samples or do iterative functions downstream. There's a parameters tab. Whatever group you're on will show you all the parameters and the samples there. This allows you to actually easily rename the stains or align parameters if you've mislabeled something. And then there's a new concept here in 11 that is called parameter sets. Parameter sets allows you to make a group of parameters that you can then focus on downstream in different contexts, say for initiating an algorithm. You always know that this subset of parameters you want to run with T-SNE and clustering. You can do that. Within the graph window, you can focus on the parameter set, and then the parameter list limits itself to only those parameters, because we're getting in this stage with even with a search bar, there's so many parameters in these files, right, that it becomes a little more difficult to manage them. There's a built-in QC algorithm that's very Flow AI-like. It touches on flow rate and stability. You can toggle on and off using the properties panel, what checks you want to do on which samples, and then click the button to apply. And the great thing about this is it runs 1 second per sample and gives you the results instead of 1 minute per sample if you're running these algorithms in R. There's a modern unmixing platform. You'll see some familiarity here where it auto-gates size and positive populations, displays that positive population in a spectral plot above so you can see what the spectral signature of the fluorochrome is. And you do your assignments similar to V10. The default is auto spill, so you only need to define the size gated population, and then it does robust linear regression. You can either choose a spectral unmixing calculation or a traditional calculation depending on your instrument. And then, of course, you can preview that matrix or create the matrix and create a copy of it, change these numbers. There is also this concept of a matrix of zeros that is called the correction matrix. So instead of modifying the rectangular spectral matrix where you don't have that one-to-one relationship between fluorochromes, you can toggle on the correction matrix, add spillover or subtract spillover to modify the interactions to your liking without actually touching the original raw matrix. And the matrices are found right here in the hierarchy of the groups, so you can drag and drop them to samples and other groups to apply the matrices without going up to a different window. I mentioned this graph gallery is really powerful, but another feature of the central plot is that we have this concept of virtual concatenation. The button for that is right here and right here. It shows 3 test tubes instead of 1, which means I'm virtually concatenating and displaying all the data at this level in the gating hierarchy from the group I'm on. So I'm actually showing 20 samples in this major plot. I'm then breaking them apart down here and showing by sample the same distribution, so I can see in each individual contributor to that. And if I wanted to, I can select the subgroups and only look at the 5 samples per group that are contributing to the total here. There is a T-button interaction context or control panel here. It shows in yellow all of the events versus the gated population, and you can control the scaling through those through that interface for all the samples that share a common compensation matrix. Here I can change, what color palettes I'm using. The resolution of the plot can be changed. I'm showing 512 here, but you can go even higher. And this properties panel, again, if you collapse all of these sidebars, can be blown up so you just have one large canvas full of individual plots by sample, by parameter, or showing the backgating and ancestry for a particular population. So it's very flexible. There is a built-in HD workflow that includes T-SNE and XShift for now. I've seen FlowSum in there, so it's coming, and UMAP, but these are not plugins. They don't reach out to R and calculate elsewhere. They're calculated natively, simplifying the setup for those of you that just want a nice, direct, high dimensionality workflow. The tables are similar to V10 and V9 in that you drag and drop a sample or a population to create which stats are going to initially be in the table, but there's no batch button. So really all I have to do is drag a group that'll tell me which samples I want to look at, and drag one or more populations that'll give me the stats. And then I can add additional statistics like means, medians, CVs, counts, and such in batch to multiple populations for multiple selected readout channels instead of doing them one at a time. So it creates this table fairly fast. You can add your keywords here. Again, this is all the panel. These are the drawers that slide out that allow you to do different things in this panel. So I've added a keyword and labeled my experimental conditions there. Now that I've created that table, I can graph it, right? I can summarize that data. And I can do a t-test or Wilcoxon rank sum test across that data to get a statistical output. And it's all linked to the original gates through the table to this chart. So here I'm focusing on a table. I'm choosing to look by experimental condition. That's the keyword discriminator that I have. And I'm showing 2 stats out of my table. That's the frequency of gamma delta T cells, which is the smaller values, and the frequency of T effector memory RA positive CD8 cells, which is showing a significant increase in COVID relative to the healthy that I've chosen to display the p-values and statistical tests for here. So linked charts is another powerful tool. Last thing, reports replace layouts. There's still a batching mechanism, which is a toggle button up at the top. You can still say, I want to look at this many numbers of rows and columns, but you can also do more fancy batching. And using this virtual concatenation button over here, I can drag in entire concatenated subsets showing all 20 samples, or I can focus on any subgroup and drag in the concatenated subgroups to show the profile of each experimental condition that I'm now graphing and charting up in the top right-hand corner. Right, so there's a lot more that you can go on here, but I am now over time. Built-in image wall support, that is imaging from your FACSDiscover instruments. You can see all those TIFFs here and change the channels. There is more analysis tools coming in the spring for this image wall, but you do have the built-in support. It automatically unpacks the CVWs and turns them into TIFFs and loads those if you just load an FCS from a FACSDiscover instrument. And finally, preferences. Getting there is the bottom left-hand corner heart, not in the top right anymore, but you have different areas. You can change the scaling, the performance settings, how much hardware you're throwing towards the application while you're using it. And then, you know, with the display settings of whether you show stains versus names, how many digits, all this stuff. And the extra benefit here is you can set these preferences at different levels. You can do it just at the workbench for this workbench and save it. Or you can say, as a user, I want these user settings. Or as a core admin for a shared computer, you can say, I want to set these at the computer level so that everybody gets the same scaling and starting preferences. That's all I've got. I'm happy to take questions, or you can reach out to me. I'm usually the guy looking at the emails at flowjo@bd.com. That's the tech support group. And if you have any comments or questions, reach out to me because I will feed that back into the development process. Anything that you give me, positive, negative, whatever, it's my job to tell the rest of the crew what your needs are. So reach out when you can. Thank you.
You're surely staying busy. Quick question.
Yeah.
Do you think that your team might consider developing color themes that are compatible for the colorblind, for different type of color blindness?
Yes, 100%. There are colorblind positive themes. We have a huge number of color palettes there in the pseudo colors. So I was using Veridis as an alternative to the standard, but there's different shades. You can do blues, greens. There's a bunch of options.
Do you know if the software is already FedRAMP compatible?
FatRAM compatible? I don't know offhand,
Okay.
but it does use GPU compute for the graphics processes. So GPU compute is more important than it was in previous iterations of FlowJo for serving up all of those images. But I would still say, you know, RAM's king, CPU processes fast you want, and GPU next. All right, thank you guys. Appreciate your attention.
Thanks, everyone. I'm going to switch gears a little bit and talk about mass cytometry, or CyTOF, talk about how you can advance clinical research with some new workflows that we have. So let me start off with this slide of an iceberg that illustrates basically the information that you can access with CyTOF. With CyTOF, CyTOF is a robust tool because you can perform high plexity, high dimensional, and simultaneous characterization of both phenotypic and functional markers at the single cell level in a single assay. And so naturally, I'm sure a lot of you have practical considerations, and there's trade-offs with using CyTOF as opposed to traditional-based, fluorescence-based technologies. But I like this image of the iceberg because it kind of illustrates how different platforms can provide you with different layers of insights. And so what we're showing here is with surface markers, you can get a really good picture of what your cells are and in some cases what they're doing. But really extending the coverage to intracellular markers and more functional markers can really enable you to open up the biology. So what we have here on the bottom are just examples of clinical trials that used CyTOF and how identification of functional markers and in some cases intracellular markers were really key in helping identify biomarkers that were linked to like drug target engagement, as well as biomarkers that were predictive of patient survival and responses. And so what enables CyTOF to do this high plexity is the fact that our antibodies, or the antibodies, are labeled with metal tags instead of fluorophores. And so the signal that comes out of it is very discrete and very well-resolved peaks. Because the instrument is measuring masses at a distinct mass, the signal, there's very minimal overlap and very predictive, minimal and predictive overlap. And so this affords us a couple of advantages, like the signal is very precise and repeatable. And unlike any other single cell technology, you know, it's not, you might run into batch effects, right, like any other single cell technology. And so one advantage to using CyTOF is the ability to freeze antibody aliquots and then stain your samples from the same aliquot because the metal tags are stable and they're not impacted. And so there's no loss or deterioration of signal. Another way to get around batch effects is the concept of barcoding, which I feel is a little bit underappreciated or maybe not as well known. And in CyTOF, this is the ability or the labeling cells with unique combinations of isotope tags. And so this can improve data consistency and minimize batch variations. You basically barcode your samples and then pool them all together prior to staining or even before acquisition. And this can minimize batch efficiencies and reduce your CVs. And so that's kind of what's shown down here with the reduced spread and lower CVs showing reduced CVs in a high abundance marker as well as the low abundance markers. So here are examples of a couple of ways barcoding has been utilized in the literature. So there's many different applications, but for example, you compare samples in longitudinal trials, clinical trials, as was used here in this phase 2 metastatic breast cancer clinical trials, where same samples from the same patients before and after treatment were paired together. You can also combine across different species or different species. So for example, in this paper, samples from PBMCs as well as spinal cords were combined together in a mouse model of multiple sclerosis and analyzed together in a single tube. And then you can also combine different cell lines or different sample types from different tissues, like shown here in this paper that did a barcoding study with mouse organoids, and they were able to multiplex up to 126 samples altogether. So not a trivial feat for analysis. And so we have a number of different multiplexing solutions. So for those of you that have already been using CyTOF, you may be aware of you're probably already familiar with our legacy Palladium barcoding kits. That's cell type agnostic, so you can use across different species. And it's basically a universal barcoding reagent. We also have antibody-based barcoding methods in the form of CD45 antibodies. And so this is specifically for if you're only interested in interrogating your immune cells. And then lately, we've been developing our TML barcoding reagents, which I'll spend a little bit more time talking about here. So TML, these are isotopologues of TML, and they are isotopes that range in mass from 122 to 133 or 130. So we have 7 different isotopically enriched tellurium isotopes. And the barcodes basically are combinations of 3 of these different isotopes as one barcode. So with a 7-choose-3 barcoding scheme, you can multiplex up to 35 samples altogether. In some cases, we have people will combine, or others have combined the TML isotopes with our monoisotopic cisplatin reagents. And so that's a 9-choose-4 scheme. And so you can multiplex all the way up to 126 samples, which is what that barcoding paper utilized. So these TML isotopes don't have any overlap with the antibody targets. So you don't have to sacrifice any channels for your antibodies when combining with this TML barcoding workflow. This is a universal reagent, so it can work on both either live cells or fixed cells. There's very minimal toxicity on cells. So it's very versatile across a number of different applications. And so just to show you an example of how we've implemented this, we created this, or we did a series of experiments comparing or analyzing together PBMCs from healthy donors along with PBMCs from endometrial cancer patients. And then we also received tumor samples from the same endometrial cancer patients and dissociated the tumor cells. And so we were able to combine all these different PBMC samples along with the corresponding tumor samples coming from the same patients. And so these samples were stimulated in vitro for 16 hours. We also incorporated IDU, which is similar to BRDU, so you can measure DNA synthesis. After the simulations, we barcoded all the samples, pooled them together, and then stained them with a cell surface cocktail followed by intracellular cocktails as well. So they were acquired on our latest instrument, which is the CyTOF XT. We have debarcoder software that can deconvolve all the data and give you individual FCS files. And then they were analyzed. And so just to show you just a couple of pieces of data, there's an app note that has much more data, but I'll just touch upon some of the key figures. And so these are OPSNI projections from the samples. And so you can see that the cell population segregated by donor, tissue, or cell types. And so notably, the clusters segregated based on similar, the endometrial cancer patients all segregated closely together. And then you can see also the clusters, how they segregated based on whether they came from the tumors or the PBMCs, and then also the cell types. And so because we were also able to profile the functional capacity of T cells by stimulating them and understanding what cytokines are produced, that's what's shown here on the bottom panel with activated CD4 T cells on the top panel, and then activated CD8 T cells in the bottom panels. And so we're looking at stimulated and unstimulated samples relating to the dissociated tumor samples, as well as the PBMCs from the endometrial cancer patients. So for the CD4 T cells, after stimulation, you can see that there's IL-6, IL-2, TNF alpha, and interferon gamma that were produced in high abundance in both the PBMCs as well as the dissociated tumor samples. IL-4 was also, you can also resolve IL-4 expressing cells, and then even the lower abundant cytokines like IL-17 and TGF-beta, you can also see clear cytokine producing cells. And these are all, again, these were all stained in the same cocktail. And then for the CD8 T cells, you can see IL-6, or sorry, IL-2, TNF alpha, and interferon gamma were produced upon activation. So we were also able to look at CD8 positive tissue resident memory T cells. That's this island that's circled here. We included CD69 and CD103 in our panel specifically to identify this specific subset of CD8 T cells. And so in the literature, these tissue resident memory T cells are help shape the immune response because they provide localized and rapid responses in the tissues that they belong to. And then also with regard to cancer, higher levels of these CD8 tissue resident memory T cells are linked with improved survival and improved survival. So there's value sort of in kind of delving deeper into this particular CD8 T cell subset. And so what we've done here is just looked at a number of different immune and activation checkpoint markers that were also in the panel. So we've got the tissue resident memory cells in orange, and then compared them to circulating CD8 memory cells in the periphery. And so you can see basically that in general, these immune activation and checkpoint markers were lower in the CD8 effector memory cells in the periphery compared to the tissue resident memory T cells coming from the tissue. So I think there's value here in terms of being able to identify rare subset populations and also understanding better like the T cell biology based on different compartments. And this would be helpful in guiding design of like immune therapy or identifying biomarkers in studies like this. And so this is just, it's a busy slide, but overall it kind of summarizes the panel that was used and gives you a feel hopefully of the modular panel kits that we have available, and also highlighting that these are validated panels that you can essentially apply to other sample types, but really also pre-validated high plexity panel that focuses on a number of phenotypic and functional markers, a number of them being intracellular as well. And then these are just some of the drop-in custom antibodies that we added. Again, I don't have the time to show all the data, but we did profile some of the tumor samples or the tumor cells in addition to the immune cells, which I've shown. So overall, I hope this showcases the ability to multiplex with CyTOF. And then we have multiple barcoding strategies. Again, I touched upon the ability to profile multiple intracellular targets. We have groups that are looking at 20 different cytokines, so you don't necessarily have to like pick and choose specifically which cytokines you are looking at. You can just sort of include them all if you wanted to, and hopefully that reveal biology that might otherwise have been missed. And I'll leave it here. Please come find me if you have any questions. I'm happy to direct you to the app note or send it to you through email. Thank you. Okay, thank you.
Hi, everyone. My name is Arpan Neupane. I'm an immunologist and a computational biologist at Ozzette. I know I'm the one, I guess, keeping you from lunch, so I'm going to try and go through this quickly. What I want to talk about today is how can you start to confidently unmix your spectral data with Ozette Resolve. I want to briefly start by looking at the general cytometry workflow, where for any given experiment, you start by generating your single color controls. The single color controls will be used to estimate the spectra, the fluorophores in your panel, and ultimately these spectra are used to unmix your data. And very often, for most users, the workflow is to leverage the bright events off of the peak detector and take that as a source of positives from which the spectra will be defined. However, as many of us have experienced, contamination, aggregation, fluorophore degradation can and does occur in many experiments. So in this example here, I've gated PerCP 5.5 off the peak detector. How can I be certain that these events are indeed a collection of true positives, devoid of potential contamination with, let's say, in this hypothetical experiment, PerCP Bio 700, or degradation product, only looking at PerCP? Importantly, how can we also understand what would happen if users inadvertently are admitting these events without knowing and carrying out unmixing with these potentially biased spectral estimates? Ozette Resolve is a new product offering from Ozzette. It is a single cell adaptive unmixing approach. And we have built in a suite of tools with Ozette Resolve that can help users interrogate and QC your single color controls so that users can identify true positives, have high quality estimates for these spectra, and generate high quality unmixing results. I'm going to walk you through a very brief example. And along the way, I'm going to address the two questions that I've posed. This is a small panel, 15 marker immunophenotyping panel. All the data was generated on PBMCs. The data was generated on Cytek 5-laser instrument at Ozette Labs here in Seattle. While this is a small panel, I think the panel could be further optimized. We do have some fluorophores that are potentially on the same cell types, namely BB630 on CCR7, C-fluor V610 on CD4, and BB605 on CD25. So we'll pay a close attention to those particular fluorophores. So I start by looking at the conventional approach for identifying positive events. Here we're looking at BB630 conjugated to CCR7 antibody. Debris, dead cells, doublets have already been removed. We're looking at the peak detector on the y-axis, B6 on the Cytek instrument. And following convention, I've added a gate at the very top here, collecting some bright events, calling them positives. Now for reference, this is what the spectra, the expected spectra for BB630 on the Cytek instrument looks like. There is a peak and B6, one in YG3, V10, and a minor peak on UV10. With Ozette Resolve, users have full access to event level spectra. What we're looking at down here is individual event level spectra for all events within this gate that I've labeled as positives. And what you're going to appreciate is that this is not true positive, collection of true positive events only, but there is two distinct set of spectra. In the white arrow, you see the true positive spectra for BB630, and in the red arrow, contamination, which also happens to fluoresce in B6. Now, before I go on to what can you do about this particular observation, I briefly want to address the second question: what would happen if I was inadvertently admitting all of these bright events as positives, true positives, into these spectral estimates and unmix the data? As a reminder, we have additional fluorophores, C-fluor V610 on CD4 and BB605 on CD25 that are similar to BB630. They're similar to one another as well. So when we unmix this data, this 15 marker panel, without doing any QC, taking all of those bright events for BB630, what you see is artifactual correlations. The unmixing is incorrect. Ozzette Technologies has built a powerful single cell query engine that can sift through large volumes of data in real time. What this means for you, for the user, is you can start to actually action on the insights that you have made. I'm going to try and get out of the pointer. So this observation that there's contamination products, you can actually action on it. You can select a reference individual event, conduct a spectral similarity search, filtering out all events that match the reference spectra that you have identified. And in doing so, we can clean up the spectra, move from this contaminated spectra, contaminated set of events, to true positive events. The spectra for BB630, after going through QC, now matches the expected spectra on this instrument. When we unmix the exact same data, this is again the same 15-color panel, we see the data are correctly unmixed. You QC, you identify true positives, you get correctly unmixed high-quality outputs. Now, this is a small panel. 15 marker panel is a small panel in this example. But we use this exact workflow even when we're working with our large panel in day-to-day basis. Our 48 marker panel called PIPO1. This is a large pan immunophenotyping panel that is identifying major cell lineages as well as functional and activation states. A very brief example of what this data looks like is at the top, we have conventional unmixing. We can see in the red arrows unmixing artifacts, and in the bottom, unmixing with Ozette Resolve, the same samples, the same controls were used. Now, in the interest of time, I didn't get a chance to talk about the other benefits of using Ozette Resolve. Maybe you did notice it as I went through the slides. We don't have any negative values with Ozette Resolve. There is no unmixing artifacts. And most critically, there is no need for post-unmixing compensation. There's an adaptive cell-by-cell unmixing solution that handles all of this in an appropriate manner. Working with Ozette Resolve is very straightforward. You start by defining a panel and an instrument. You upload your FCS files. You define a control set. These are the set of files that you'll be using to unmix your full-stained data. QC, as I've done here before. Unmix, and then you can either move it to the Ozzette analysis side, either Ozzette Discovery or Ozzette Endpoints, or you can download the data off of the platform and use it on any cytometry analysis software. If you would like to try out Ozette Resolve, you can come find me. Also, I'm happy to take any questions. So the method is relying on the empirical spectra that you have generated. So the method relies on empirical spectra that you have generated from your single color controls. At present, you are referencing a database to know what the reference, the expected spectra should look like, but we are working to include that in the workflow itself, so you don't have to go out to look for that spectra. Yeah. I think that's it. Thank you so much.
Okay, so that concludes our lightning talks.